yeah, I'm Max. I'm still a master student in neuroscience, and Carsten is supervising my project. And I'm particularly interested in fMRI reliability to bring fMRI towards the clinical usage and yeah, make it a method which tells us more reliable about the brain. And yeah.
Yeah, my name is Mengyun Wang, and I'm quite new to this, to MMIV. Actually, I got here like 3 months ago, and I'm part of Carsten's group and working as a postdoc. My research field mainly focused on resting state, fMRI, and brain simulation. So maybe we can start our presentation. Okay. First, I would like to thank Frank for inviting us to give this talk. And today we would like to share this an ongoing project. We named this project Bergen Breakfast Scanning Club. In this project, we would like to scan ourselves for 1 year, and would like to tracing down how our brain changes along this 1 year. So this project was initiated by Max, me, and Rune. So here is the outline of this presentation. First, I will share with you the background to share with you what kind of research inspires us to do this project. And then Max will share with you our motivation to do this project, why we do this project, and how we're gonna do this project. So if you are familiar with fMRI studies, you're probably familiar with this procedure. First, you need to recruit dozens or hundreds participants to your experiment, and you scan them, you got the data. Depends on whether there is a task or not, you will get a task related, or if there is a task, you will get a task related fMRI data. And then you analyze it, you get a brain activation network for this specific task. And also without task, you will get resting state fMRI data. And you can also extract very useful information, such like this resting state brain networks. With this kind of method, we can generate some general information. It means that on group level, we can say that which brain region or which brain network is responsible to which kind of cognitive functions. I call it, generally speaking. But how about this specifically speaking? How about the individual details? So one way to achieve or one way to look at individual details is like this way. Instead of scanning dozens or hundreds subjects with very little time, we can intensively scan several subjects with a long time period. In this way, you can also get similar data size, and also you can tracing down each one of your participants' brain changes. So you may wondering the sample size. You may wondering, several subjects couldn't be not enough, and could you extract useful information from just several participants? The answer is yes. And actually, this intensively scanning several subjects was nothing new. Back in 2017, there is an article published in neuron. And the title is Precision Functional Mapping of Individual Human Brains. In this article, they actually used this kind of intensively scanning several subjects to tracing down individual details. And this project led by Professor Nico Dusenbach. And in this paper, they found that the individual brain organization is quantitatively different from group average estimates. And also they found that the individualized measures of brain function become reliable with large amounts of data. And last, they also found that individuals exhibit distinct brain network topography and topology. So if we would like to intensively scanning someone, we need constant fMRI available time slot. But we all know that in daily life, in daytime, fMRI machine is always fully booked. But at midnight, the fMRI machine is always free or available. So Professor Dusenbach thought that they can use that kind of time slot to intensively scanning their subjects. So in this paper, they actually scanned their participants in midnight. So they named their project Midnight Scanning Club, and they came up with this very cool logo. And we read this when we read this article, we thought, scanning subjects at midnight is not making any sense, because it's not a representation of ordinary people's daily life. So we think that we probably could intensively scan several subjects at the daytime. So we named our project Bergen Breakfast Scanning Club. So back to this article, you can see that they just scanned 10 participants, and they collected the data in 10 consecutive days, and they have 3.5 hours structural fMRI data, and 5 hours resting state of functional connectivity MRI data, and also 6 hours task-related MRI data. So there are lots of results in their in this publication, but in here I just highlight these results. We can see here on the group level. By the way, the red color is represented as the DMN, the default mode network. So in this example, on group level, we can see that seed A and seed B are both belonging to the DMN network. But if we look at the individual level, specifically like this number is 6. So we can see that actually seed B is not belonging to the DMN. Actually, seed B is belonging to Celine's network. So what we take away from this article is that group level brain networks could blur the individual character variations. And also some of the networks are not consistent across all subjects. For example, in this example, the DMN network. So in this article, they just scanned the subjects 10 days. It's like a very short longitudinal study. How about scanning subjects for more longer time? And this article I would like to present is using this kind of idea. This article was published in 2015 and in Nature Communication. The title is Long-Term Neural and Physiological Phenotyping of a Single Human. So from the title, you can see that he just scanned just one subject. And this project was led by Professor Russell Podrick. Because he scanned himself, so he named this project My Connectome Project. So you can see the timeline. He scanned himself almost 1.5 years. So the idea behind this project is that back in 2010, there were lots of resting state fMRI studies were coming up, and Professor Russell Podrick was at that time mainly focused on task-related MRI, and he was very skeptical about this one-time snapshot of resting state fMRI. So he thought that is this reliable? Is this the validity of this kind of one-time snapshot of brain can provide such rich information? So in order to find out the reliability of resting state fMRI, so he scanned himself for this long time. So you can see that he mainly focused on resting state fMRI. He scanned himself twice every week, and he got 100 sessions, resting state fMRI sessions. And also he collected structural MRI, but I think he scanned himself just once a month. And also there are lots of results in this publication, but I would like to highlight this one. Back to his question, back to his curiosity that whether resting state fMRI data is reliable from long time period perspective. So he found that from figure A, we can see that within subjects, actually the lower cognitive processing regions, such like visual, somatomotor, and dorsal attention regions, showed a very high variability. But in contrast, between subject, this figure B results are extracted from open database from the HCP data. So between subjects, he found that actually the higher cognitive processing regions, such like frontal, parietal, cingular, opercular, attentional, and default mood regions showed a very higher variability. So he just found this kind of phenomenon, but he didn't provide any explanation or some kind of reasons for this. So we think that we think that if this is just a one case thing, or it can show in many subjects. So in our project, we would like to verify these results. So that's the first part of the project. I would like to just share with you what kind of research inspire us to do this project. From the second part, Max will take over. Max.
All right. Yeah, so as Mengyun already said, that there are some major limitations to the first two studies he presented. And yeah, as he said, the main thing with study one, the one where they tested 10 individuals always at midnight, obviously has this problem that at midnight, the circadian rhythm might introduce some differences to the signal, which you would not have in a normal day, and then also not find in the data of most studies because they're not obtained at midnight. And then Russ Poldrack's study, the second reference, was always, the data was always recorded at 7 a.m., which is maybe also not the most normal time. So we thought maybe we could find something in between. And there was also the idea that we could actually recycle some slots from another project. So that fitted us very well. So that we came up with 10 o'clock slots, and we would but also like to introduce some new slots now, which are in the afternoon or evening. So, but yeah, we found the middle ground between 1 person and 10 people, and between 10 days and 1 or 1.5 years. So we thought, what if we do the same, more or less the same thing as Russ Poldrack? So we scan 100 times within around 1 year, and then it's Rune, Mengyun, and me who are going to get scanned. And yes, since there is a lot of discussion about the variability of the fMRI signal, and nobody knows exactly what this is to be contributed to, we also want to observe a lot of different factors to get a better idea of where this variability comes from. And yeah, this will definitely not only help to replicate or maybe even verify those previous results, but maybe also to observe some additional factors which we might have to look at when doing fMRI studies. And what we're especially interested in is a time of day effect. So this is why we want to scan in the morning and in the evening or afternoon. And then some seasonal or time of year effects. So there's a lot of stuff going on with light, and especially in Bergen. Maybe we should also look at the weather and how clouds shadow the city, and so just to see if there's maybe an effect on us and then our connectivity in that sense. And we will also have a sort of manipulation in our study, because Mengyun is learning Norwegian at the moment. And so we're going to do some language tasks as well. And we have the advantage now that Rune is a native speaker. I have acquired Norwegian like maybe 1.5 years ago, and I would say I'm maybe on a C1 level, and Mengyun is just learning it. So then we can maybe even compare something there, although this will be more like a pilot test. And additionally, we will also look at inflammation markers, different hormones like melatonin and cortisol. We'll record mood, our sleep, exercise, food, medication, alcohol intake, and also caffeine intake, both every day and each time before scanning. And this is more or less what the protocol looks like. So every morning when we get up, we answer some questions about sleep quality, and how many hours we slept. So that's basically retrospectively from the last day, because it can happen that you're on your phone and do the questionnaire, and then you get up again. And so we thought we do that in the morning, and then in the evening we will record things like how much you were eating, how much you were exercising, gut health, stress, and so on. So those we do every day for probably 1 year. And then in addition to that, when we come into the scanner room, we're gonna do some blood spots. Oh, I have to hurry up, okay. Yeah, we do some blood spot and saliva testing and take blood pressure. And while we're gonna get scanned, we do a heart rate and respiration, and also some mood questionnaires. Okay, now I try to do it as fast as possible. Let me see what I have next. Yeah, so we have now 3 different protocols. One is basically a structural protocol where we do all kinds of structural measures. Then we have so that's this one. Then we have a resting state, mainly focused on resting state session, and we have the task session I talked about, where we assess the language. And so just to give a quick roundup. So in the end, we want to connect all those factors to structural and functional measures. So how is our circadian rhythm working? This is influenced by our hormone levels and light and illness and exercise and so on and so forth. So I hope that was not too rushed, but yeah, that is mainly it. And then we hope to get through this 1 year without any complications.
That's fantastic. Thank you very much to the Breakfast Club. It was a great, really interesting talk, and I think it's cool the protocol that you've designed and all the things you're thinking about. I think that will really provide some amazing and breadth of data. And I noticed that that the guy who's just only scanned himself got himself into Nature. So I hope you're going to aim high. And in the interest of time, if anyone has questions, please write them in the chat, and we'll have a go around at the end. But in the interest of time, we're just going to lead on to the next talk, which will be introduced by Sherin Sugathan, who has taken over this year. So he's new. Welcome to Sherin. And Sherin will be introducing the next speaker.
Thank you, Frank. Hello, everyone. I am very pleased to welcome our next speaker, Stian Soltvedt. Stian is a master's student at the University of Bergen, and he works on a vascular visualization project for medical education currently. And for this session, Stian is going to present the Open Anatomy Explorer, which is a web-based viewer for studying anatomy. And I've seen some part of this project already, and I already love it. So I'm sure that you are going to enjoy too. So yeah, over to Stian.
There we go. Thank you. Hi again. I'm Stian. I'm a master student at the University of Bergen, studying medical visualization, and this is the Open Anatomy Explorer. It came into being as a result of the current situation around teaching anatomy, which is an essential skill for most medical professions. And at the University of Ghent, they were doing dissections on donated bodies in order to give the students a deeper understanding of human anatomy with the actual specimen. But between the practical limitations, oh dear. I blame the cat. Well, that wasn't great.
That's okay, no problem. I think a lot of us have cats too, so...
Yeah, he's a bit of a problem. Yeah, and between the practical limitations on how many students can participate in such sessions, the ongoing pandemic has... Stop! And the ongoing pandemic has also provided additional challenges with these teaching activities, problems moving that to a virtual format. Together with the Ghent University Hospital, our contribution to anatomy education is this Open Anatomy Explorer, which is intended to be an accessible web-based viewer for studying anatomy. And in contrast to existing solutions, which are frequently based on meshes designed by artists, our goal is to use real surface scanned specimens with realistic shapes and textures, since that's a lot easier to do these days, since you don't require a lot of people to scan single specimens at the time. And that gives us realistic shapes and textures, with close to what you may see when working in the field. And yeah, we want this to serve as an educational platform available to access by anyone with a modern web browser, and see where we can expand it from there. And this project is a follow-up to the online anatomical human project by Smith et al. In 2016, which was successfully used and deployed in a massive open online course to teach the anatomy of the pelvis to students worldwide. And with our project, we hope to continue to explore this problem space by supporting a wider variety of these surface scan specimens, together with a comprehensive set of labeling and quizzing tools. and has a little taste of where we're at. So yeah, the first part of the project is, of course, letting creating a tool set for them to label these scan specimens. So we have essentially a painting tool, which allows people to label these modules with different colors and transparency to highlight and label different features and show and hide them and all that stuff. We also provide a service to store all these to make them easy to share. We can create collections of labels to browse through, as well as like quickly load different bookmarked label sets and their related models to browse through the various labeled models. From there, we have worked on a flashcard quizzing system, which intended to assist with space repetition of anatomy. We have location questions. Where is this region or that region? Where you're asked to click on them. And we also allow you to shuffle questions and some miscellaneous features like that. And another question may be like, what is the name of this region? And then it will place a little light widget on it to highlight it and move the camera there to make it clear which region's being asked. Come on. A quick note on labeling. The current we attempted some different ways to label things before settling on just painting straight onto the models with vertex labeling. We also tried placing 3D models or spheres to mark places of interest. We've tried painting directly on the texture, which, if you can see this texture of the arm, is not a very good idea because the texture space does not really correspond to real 3D space, and you can't just paint straight onto the texture, even though it was quite space efficient. So in the end, we're painting individual vertices, which is quite fast, but requires a bit of memory. But since the models are a bit compressed to avoid the website taking loading for 30 seconds, we found it's fine in practice. Currently, we're at the stage where we're going to take it into use soon. We hope to get students working on scanning and labeling in this month or next month. It's somewhat useful for quizzing and flashcards. We haven't got to deploying that yet, but we're hoping that will be useful. It's not quite ready for examination use yet. We've just got some basic user support in, and if we could get some integration with examination tools and some additional security things that we'd require for that, we hope to maybe do that sometime. We also want to add some more question types. Orientation questions would be nice, like where's the posterior, anterior, and the top and the bottom of the various bones, like the tibia. That would be nice to support. And we would also like to give it a bit of a more compendium, lookup research tool usage by letting you link the labels to various information sites to make it easier to browse through the various models and read more about the features in question. And sometime we would really like to also CT scan these specimens so that you could also see how slice views from a CT scan would look like for this specific specimen, to get an impression of what you're looking at there and help see what's what in there. Maybe even do some volume rendering by that, which would be very interesting. We think that would be useful as well for getting a proper feel for what's inside. And of course, the aforementioned examination mode would be much appreciated by at least the university prep. And that's the main part of my presentation. Yeah, I assume we're taking questions afterwards.
If we don't have, we would like to thank to all of the speakers for great presentations. We would like to thank you as well, Heidi, Havjin, and Eric for their hard work in the seminar committee. and welcome Kari and Sherin. And we are inviting you for the next seminar that will take place the 12th of the March, and hopefully we can be able to meet at least personally in the auditorium. Thank you very much.